Hi everyone, today we will be looking at cannabis as a potential source of natural medicine. This presentation has been brought to you by Mariam, Cecilia, Dean, Ria, Zara, Isra and Liam. Seriously Liam, you had one job. Let me just quickly remove this. Right, here's what I wanted. This is the more commonly known form of cannabis, which is also known as pot or a joint.
And here's our overview slide. So basically this is what we're going to go through this presentation, so stick around with us for the next 10 minutes or so. So cannabis is sourced from the cannabis tree, and we've got all three common types written here for you as well. Sativa is the most commonly known one in Western culture, which gives you the high, whereas indica at the top here is also known as sedative cannabis, because it makes you drowsy.
There's ambiguity surrounding when cannabis was first used, but written records suggest that in ancient China, Emperor Shen Nong, also a renowned pharmacologist, cultured a form of cannabis known as hemp for therapeutic treatment of more than 100 ailments. More recently, cannabis was listed in the American Pharmacopoeia for around 100 years, up to its removal in 1942. The reason for this was that it was being sensationalized in pop culture and media as a psychotropic drug for recreational purposes.
Yeah, that's cool, Cecilia. But even in the 100 years that it was part of the US Pharmacopoeia, it really cemented its place as a really potent analgesic. So if you have a look here, people are using it for tooth pain, they're using it for joint pain, and during childbirth as well, which makes it all the more reason that it should be considered as a potential analgesic.
Antispasmodic and anti-tremor effects of cannabinoids have been demonstrated in an autoimmune model of multiple sclerosis. Cannabinoids have also demonstrated neuroprotection by slowing down the rate of neuronal degeneration in multiple sclerosis model and improved memory in rats with induced memory impairment. Cannabinoids have long been known for the analgesic effect, although shown to be effective at reducing pain in conditions such as cancer. A major hindrance in the side effects, which include cognitive and motor impairment and psychotropic effects. Cannabinoids have been approved for their indication in cancer and AIDS-induced anorexia and cachexia, as well as chemotherapy-induced nausea and emesis. Currently marketed synthetic cannabinoids include dronabinol and nabilone. The anti-inflammatory effect of cannabinoids was demonstrated in non-obese diabetic mice with hyperglycemia-induced inflammation. Cannabidiol and delta-9 THC were demonstrated to be effective in reducing glutamate toxicity seen in Huntington's disease, suggesting an antioxidative effect. Cannabinoids target two main receptors, cannabinoid receptor 1 and cannabinoid receptor 2. Once activated, these receptors can affect different functions, including pain perception, mood, appetite, and memory. CB1 receptors are abundant in the brain. They are generally concentrated in areas associated with nociceptive perception, including the thalamus, central gray matter, and the amygdala. CB2 receptors are mainly associated with peripheral effects and involved in the inhibition of pro-inflammatory factors released by non-neuronal cells, like immune cells. CB1 was first validated as the target for cannabinoids in 1988 with a ligand binding assay using a radio-labeled CB55940 ligand, which is a cannabinoid of delta-9 THC. The assay indicated rapid binding of the ligand to a receptor concentrated on synaptic terminals of neurons. The receptor was soon cloned in sequence and was determined to be a 7-transmembrane G-protein coupled receptor. In 1993, complementary DNA library was produced from the macrophages. One clone, now coined CB2, had 44% sequence homology to CB1. Validation study was conducted by integrating the receptor gene into the plasmid SC36, then inserted into expression vector CDM8. This study showed that some cannabinoids have greater affinity for one receptor over the other, while some demonstrated similar affinities to both.
What we want is to obtain the concentrated extract referred to as cannabis oil, and you can get this from the leaves and the buds. We can obtain this in one of four ways, which we've listed here. So we've got either the olive oil extraction method, we've got dry sieving, ethanol, or carbon dioxide extraction. The last one we're going to be looking at a bit more because it's actually quite commonly used. So with the carbon dioxide extraction, you end up with a really pure and potent oil, which is isolated. So in one end, you throw in your buds and your leaves, and these are subject to extremes of highs and lows of CO2 pressure. Unfortunately, this method is quite expensive and there's quite a steep learning curve.
The benzopyrene ring in delta-9 THC is an essential requirement for activity, as the open ring significantly reduces activity seen in the case of cannabidiol. However, the benzopyrene ring in delta-9 THC alone cannot confer activity, since cannabichromene, which possesses only this one functional group, is inactive. The oxygen in the benzopyrene ring can be substituted by nitrogen without loss of activity. Attachment of a non-planar allocyclic ring to the benzopyrene ring in the 3-4 position is important for activity. However, a planar ring attached, as seen in cannabinol, causes knee inactivation. This allocyclic ring can be substituted by a heterocyclic ring or a bulky substituent at the 4 position without loss of activity. A range of substituents can be introduced to the allocyclic ring while still retaining activity. This means the methyl group can be replaced by hydroxyl, hydromethyl, or ketone group. The length of the aromatic side chain can be varied without loss of activity, but must be a minimum of 3 carbons to retain activity. Increasing the length of the side chain with branching enhances potency by several folds. Substitution of the allocyclic methyl group by hydroxyl group allows delta-9 THC to retain potency. Moving the double bond of the allocyclic ring from the 9 position, or increasing the aromatic side chain to 3 carbons, reduces potency of delta-9 THC, and hydroxylation of the 8 position can reduce potency by 80%.
Synthetic derivatives of cannabis used for medicinal purposes include dronabinol, which is used in anorexia and symptomatic relief of neuropathic pain, canosol, which is used as an antiemetic and in glaucoma, and also nabiximol, which is used in cancer-related pain. Another well-known derivative is nabilone, which is commercially known as Sesamet and is used primarily to relieve nausea and vomiting in cancer patients. It is derived from the THC molecule and has undergone structural alterations to optimize drug-target interactions. This includes substitution of the methyl group here with a ketone. Addition of the ketone allows hydrogen bonding, where the carbonyl oxygen acts as a hydrogen bond acceptor. It is also able to form a dipole-dipole interaction with the binding site. If alkyl groups interact with a hydrophobic binding pocket, varying the bulk of the alkyl group as shown here allows one to examine the dimensions of the hydrophobic region, potentially increasing the interactions and hence the potency of the drug. One of the main chemical constituents of cannabis that acts on the brain is delta-9 tetrahydrocannabinol, otherwise known as THC. Others include cannabinol, cannabidiol, cannabichromene, and cannabigerol. THC is responsible for the psychotropic effects of the drug and mimics the action of endogenous cannabinoids, but when inhaled, it circles the body in much larger quantities than needed. Endogenous cannabinoids are naturally occurring neurotransmitters found within the body and are known as anandamide and 2-arachidonoglycerol. There are 2 main ways to take cannabis. You can eat it, where it will take longer to have an effect, or you can inhale it for a faster onset of action. Upon ingestion or inhalation, the exogenous cannabinoids enter the bloodstream through the digestive tract or across alveolar walls within the lungs. They then migrate towards the CNS, whereby they are able to cross the blood-brain barrier and exert their effects on the CB1 and CB2 receptors present within the brain. The influence of cannabinoids on pain transmission is shown in this comparison of normal pain transmission, the effect of endogenous cannabinoids, and finally the effect of exogenous cannabinoids. The cannabinoids bind to presynaptic receptors of the neurons, and a G protein cascade is activated, resulting in a decrease of calcium influx and neurotransmitter release. This ultimately dampens the signals that would be recognized as pain by the brain, resulting in the analgesic effect of cannabis.
Interest regarding the use of medicinal cannabis has increased significantly in the past few years. Despite this, there are 3 main factors which still hamper its progression: a lack in scientific knowledge, how readily it's accepted by society, and whether it still fits within the scope of the law. Hopefully, though, it will be considered as a potential source, as the risk of opioids is much greater than the risk of using cannabis. So here are the faces of the 7 beautiful people who presented this to you today, and here's a list of all the stuff that we did. Thanks for watching.